Hello, my name is Atishay Jain, and I'm a PhD student in the Department of Computer Science at Brown University, and I'll be presenting our work on clustering spatial transcriptomics data with nested graph neural networks. Let us start with what spatial transcriptomics is. Spatial transcriptomics is a technology that allows us to measure gene expression of tissue locations or spots alongside the spatial information within the tissue. Clustering spatial transcriptomics data allows us to annotate cells and better understand tissue structures and disease pathology. Existing spatial transcriptomics clustering models generally train on graphs that are created using spatial information, but they do not use any gene-gene relationship graphs. Since genes interact and are expressed differently in distinct cell types, we hypothesize that including these gene-gene relationship graphs will help improve clustering performance. So how do we integrate these gene-gene relationship graphs and spatial information together? That's where our nested GNN framework comes in. In the first step of our framework, we use k-means on the spatial information to generate groups of spots, called spot groups. We then use the gene expression matrix to generate gene co-expression matrices for each spot group. With these gene co-expression matrices as edges and these gene expression values as node features, we generate spot-specific gene-gene relationship graphs. These gene-gene relationships graphs are then passed through our first GNN, which we call the inner GNN. And for each graph, and therefore each spot, we get an output. These inner GNN outputs contain values that represent both the gene expression values as well as the gene-gene relationships. Then, with spatial information as well as the inner GNN outputs, we generate these spatial graphs. The edges are from the spatial information, while the node features are from the inner GNN outputs. These spatial graphs are then passed through an outer GNN or second GNN, which we call the outer GNN, whose aim is then to reconstruct the original gene expression. Therefore, with our framework, we generate embeddings that contain the information, the gene expression information, the spatial information, as well as the gene-gene relationship. And then we use these embeddings to perform clustering. So now that we know our framework, let us check at how well our model performs. As you can see, so in our experiments, we tested our model against the state of the art, which is GraphST, and we tested it over two data sets. The first data set consists of 12 human dorsolateral prefrontal cortex samples, and the second data set consists of 5 mouse preoptic hypothalamus samples. In the first row of results, you can see we have the ground truth, GraphST performance, and nested GNN performance. This is on the first data set. As you can see, our nested GNN is able to identify the small cluster that the GraphST is not able to pick up. And therefore, visually, we get a confirmation that our model is able to use spatial information and gene expression and gene-gene relationship information to perform clustering, to do better clustering. In the second data set, as you can see in these three images, again, with the ground truth, the GraphST output, and the nested GNN output, we can see that the nested GNN is able to closely identify most of the clusters, and it's symmetric. Whereas in GraphST, you can see that some of the clusters are kind of all over the place and are disjointed. And therefore, we get a visual representation of how well our model performs on these data as compared to the state of the art. To go into quantitative analysis, we have two graphs, one for the first data set of the 12 human samples, and the second for the data set with the 5 mouse samples. As we can see in both the graphs, our nested GNN seems to have a higher overall ARI over all the data sets. And we also note that our nested GNN has a 6.9% and a 6.3% increase in the adjusted rand index or ARI score over the GraphST in both the data sets respectively. While our model has shown impressive results and better results in the state of the art, there are still limitations and open questions for the future. The first and main limitation of the model is that since it is a nested structure, the model ends up having a lot of edges in both the layers of the model. As a result, we require a lot of GPU memory to train. For instance, a few of the samples in the human dorsolateral prefrontal cortex dataset required 24 GB GPU memory to train. This can be offset by reducing the number of edges in the data, but that could lead to a drop in performance. Secondly, we've only tested our model on 2 datasets. We'd like to prove its generalizability by testing our model on other datasets as well. And for the future, since our model learns gene-gene relationships during training, we aim to use the model to identify important cell-specific gene-gene relationships. This is uh, an important part of the problem which we plan to focus on in the future. Thank you for listening to our presentation.